We're very happy to welcome Professor Muyinatu. So I was reading about her career, and it's really something very impressive, very inspiring. She's currently at Johns Hopkins, where she's the Joan C. Malone Associate Professor of Electrical and Computer Engineering, Biomedical Engineering, and Computer Science. There she founded and she directs the Photoacoustic and Ultrasonic Systems Engineering Lab. So the aim of that lab is to engineer and deploy biomedical imaging systems, mostly for diagnostics and general unmet clinical needs. I was having a look at some of her papers and, being very keen on the whole imaging inside the body. She's really pioneer in what is deep learning in ultrasound imaging formation. She has some very cool work on imaging blood vessels in the pancreas and liver, computer vision, and so on. So photoacoustic itself is a wonderful technique, as we know. She earned her bachelor's degree in mechanical engineering from MIT. She then got her PhD in biomedical engineering from Duke University. And then she was a fellow from the Whitaker International program, where she went to the UK to do, so it's basically targeted to biomedical engineering for placement overseas. She then did a postdoc at the Engineering Research Center for Computer Integrated Surgical Systems and Technology at Johns Hopkins, where she's now. She's also Associate Editor-in-Chief of several of the Institute of Electrical and Electronics Engineers journals. She's a member of several scientific advisory boards, including CZI, which I was also very excited about, in general related to what is photoacoustic-guided surgery and deep learning for beamforming. And she has been recipient of many, many awards, which is also, very, very exciting, very inspiring. This includes the MIT Technology Reviews Innovator Under 35 Award in 2015, and an SF Career Award, an NIH Trailblazer Award, and amongst the most recent, so Maryland's Outstanding Young Engineer Award, and among the most recent is the SPIE Early Career Achievement Award and the IE Ultrasonics Early Career Investigator Award. So really excited to hear your talk, and yeah, very keen. So yeah, the floor is yours.
Thank you so much. That was such a lovely introduction. I appreciate it. And, I'm really excited to be here to talk to all of you about how we in my lab, the Photoacoustic and Ultrasonic Systems Engineering Lab, which is abbreviated as Pulse Lab, how we are listening to the sound of light to guide surgeries. So without further ado, I want to first, give you a broad overview of the lab before I delve into this research topic, where in the Pulse Lab, we follow this cycle that you see here, where we develop theories, models, and simulations, such as this optical simulation of prostate brachytherapy seeds within a prostate to understand how light dissipates around the seeds. And we use this knowledge to design things. We design what's called beamformers, which are signal processing techniques. And we also design imaging probes. Beamformers require a lot of mathematics, and more recently, we are replacing the mathematical models with deep learning models. And we also design physical objects, such as this imaging probe showing a side-firing light delivery system for dissipating light to visualize brachytherapy seeds. Our designs, especially the signal processing designs, are interchangeable between photoacoustic, abbreviated as PA, and ultrasound, abbreviated as US. Ultrasound and photoacoustic imaging systems can use similar software to achieve our overall goal of building and testing prototypes that improve both ultrasound and photoacoustic image quality. And then we take these designs on the hardware and software end that are backed by our theories, models, and simulations, and we integrate them with commercially available systems, such as an ultrasound system, a laser, or a robot to together create a new system that's the first of its kind to address a clinically relevant problem. And then we might take our systems to the actual patients who could benefit from them and engage in clinical studies to see how these systems that are the first of their kind can be used to improve healthcare. And then we might learn something from our designs, which would require us to revisit our theories, models, and simulations, and the cycle continues from there. So I'm really excited to talk to you about one aspect of this cycle, and I'll start and motivate that aspect by asking the question, how exactly can we listen to the sound of light? Well, we can take cues from nature and watch this video that shows an amazing sight that I saw on one of my many flights around the world. It's the view of a thunderstorm from an airplane window. And as you see and watch this view, you can appreciate several parallels to my research. The first being the answer to the question, how we can listen to the sound of light. We know that when lightning strikes, thunder is sure to follow. And that's because the light separates the air surrounding it, causing it to rapidly expand and contract and generate that loud roaring boom that you hear when you hear thunder. And this conversion of optical energy to acoustic energy is very much related to the photoacoustic effect that we take advantage of in a much smaller scale inside of the human body. And this is how we can listen to the sound of light. And we do that with this setup shown here that merges an ultrasound system with a laser. And together this creates a photoacoustic imaging system. And if we want to image a photoacoustic target, such as the one shown here, we transmit light to the target, which absorbs the light, undergoes thermal expansion, and generates a sound wave. The initial pressure distribution of that sound wave is governed by this equation that you see on the slide. It says that pressure is proportional to mu sub A, which is the optical absorption of that photoacoustic target, gamma, which describes the thermal properties of the target, and phi, or the fluids, which describes the properties of the light that was transmitted to the target, the energy of the light divided by its area. So if I have a sufficient quantity of any one of these variables, I can generate that initial pressure distribution that would then allow me to listen to the sound of light. And I listen with conventional ultrasound transducers that are interfaced with the body to create ultrasound images that you might see in a clinic today. And then this ultrasound transducer contains piezoelectric elements that convert the mechanical pressure waves that are sensed into electrical signals, which are then sent to an onboard computer for processing. And the first step in the processing chain is beamforming. It's one of the key components of my lab in that big cycle that I showed you earlier. It can be considered as the first line of software defense against a poor quality image. So if I have a sufficient beamformer, then I have a sufficient image. If the beamformer is poor, then my image will be poor. Once I get the beamforming right, I can apply some additional post-processing, such as scan conversion, log compression, and filtering. And finally, the image is ready for display on the monitor. And of course, since I'm using standard ultrasound imaging hardware to get this image, I can still use that information and interleave it with the photoacoustic information that's provided by the transmission of light and improve the overall experience and get both ultrasound and photoacoustic images together, as you see in this example shown here. From a historical perspective, photoacoustic imaging has made an impact and has shown remarkable promise in several applications. It started out as this nice, neat tool to enable small animal imaging and unlock the mysteries of the brain, starting with what you can see here as an image of the vasculature of the mouse brain. It then transitioned on to show how it could be used as a potential indicator of cancer, which we know can recruit blood vessels due to the angiogenesis process. And so you're seeing an example of the demonstration of that promise by visualizing the photoacoustic image of blood vessels within the palm of this person's hand. Photoacoustic imaging can also be used as a molecular tool to determine the uptake of a drug over a period of time to determine if the drug is going to a targeted area as intended. So in the example on the far right, you can see immediately after injection or before injection, there's no photoacoustic signals. And then after injection of some molecular agents, you can see the photoacoustic signal more clearly. A fourth example is shown on the bottom, where you can do intravascular imaging in small, tight spaces, where you can send an ultrasound probe and an optical fiber pair into the lumen of vessels and spin it around and create an ultrasound image and a photoacoustic image. And in this case, the photoacoustic image is an image of a stent, which is made of metal and has a high optical absorption, even higher than blood vessels. And so you can see the metal stent and overlay that on the ultrasound image of the blood vessel lumen, and determine if the stent is rightly sized for the vessel. Now, the novel application and design introduced by my lab over the past several years has enabled this technology to be used and shown as a potential utility and promising technique for surgical guidance. And in order to do this, we had to introduce a novel set of designs where the traditional designs have light sources that are either attached to the ultrasound probe, as shown in the bottom example, or the light source is located at a fixed distance from the receiver, as shown in the upper left example. The designs introduced by my lab break this rigidity in system design and add more flexibility by separating the light source from the ultrasound probe and instead attaching it to a surgical tool. And one reason we do this is because, as you can see, just like you can see the vessel, the image of the stent in the vessel, and the stent is made of metal, similarly, surgical tooltips are made of metal, so they create a high photoacoustic signal in the image. And we want to use this image of the tooltip to determine how closely it is related to a blood vessel of interest that we don't want to injure during a surgery. But this blood vessel could be hidden by tissue, so the surgeons won't see it very readily. And so both the tooltip and the vessel can create a photoacoustic signal. They absorb the light, undergo thermal expansion, and generate a signal that's sent to the ultrasound probe. And that information can be used to guide surgeries. So here's an example of one of the first designs created to enable this new field of surgical guidance, photoacoustic-guided surgery, where we rely on multiple simulations, such as this optical simulation that tells us how many light sources or optical fibers should surround our surgical tool. And we can also get information about how well the tool, the video is not playing, but if it were, you could basically simulate what the profile would look like as a function of distance from the tissue surface. So we get this shape very close to the surface, and we would see the simulations predict this type of shape farther away from the surface. And this first working prototype was demonstrated for neurosurgery, where in this surgery, I hold a prototype and enter a mock surgical environment. And the two black lines that you see represent internal carotid arteries, or vessels that we don't want to injure. And you can see how close the tooltip is relative to these vessels that I don't want to injure, just as I'm navigating around. And you can appreciate this linkage by looking at the videos, but in a real surgical environment, the structures that we want to avoid are hidden. And so we added a layer of complexity to the mock surgical setup. And on the right, you would be able to appreciate how well the technique works even in the presence of bone. This was previously a video, but it looks like the video is not playing right now. And we can see, we would basically see that the same information is conveyed even in the presence of bone. Now, this concept of photoacoustic guided surgery was initially introduced for neurosurgeries, which are minimally invasive surgeries produced through the nose. And you can appreciate that the surgeries are done with small tooltips, and you can attach small optical fibers to the tooltips to generate that photoacoustic response and have it be sensed by an external ultrasound probe shown here on the temporal region of the skull. But more recent work from my lab shows that there are multiple acoustic windows that could receive the generated sound, such as the eye or even in the nose if the probe is small enough. Branching out from this initial idea, we realize that it's not limited to neurosurgery. There are a host of other surgeries that could benefit, such as surgeries performed with minimally invasive robotic instruments, surgeries performed to correct spinal deformities, surgeries performed to remove the uterus, as well as robot-assisted biopsy needle guidance, which is more of an intervention rather than a surgery, such as similarly cardiac catheter procedures are also an intervention, and liver surgery can also similarly benefit from the general idea. So I'll talk now about some implementations that are highlighted here. I'll go into 3 or 4 examples, And then I'll talk about some of the challenges that we are currently exploring in my lab, introduced by this new class of imaging technology. And I'll talk about one of the more promising examples to enable this technology by moving away from rigid transducers like the one that you see in the neurosurgery example, and allowing our ultrasound sensing to conform to the multiple different shapes and sizes that you see here on this slide. And then I'll end with a summary and outlook. So the first implementation I wish to share with you today is how we can integrate this technology with a Da Vinci robot. And if you're not familiar with the Da Vinci robot, it's a way to provide teleoperative surgery where the surgeon would sit at a master console and the subtle motions of the surgeon's hands translate to motions of the robotic arms, which are holding surgical tools that can be inserted through keyhole-like incisions into the body. Our goal with a photoacoustic approach is to bring in the photoacoustic system and attach our specialized light delivery systems to the surgical tools to enable the additional visualization of structures that are hidden. So we now transition to a view of what the surgeon would see when looking through the master console and navigating this tooltip around the area of interest to determine where to make an incision. The inset shows a photoacoustic image video that could be displayed through that master console. And in this video, we see that there's some photoacoustic signal structure when the surgeon moves close to one region in particular, and then that structure disappears as the surgeon moves away. And so this could provide information about where to either target if that's the structure of interest, or avoid if that's the structure we don't want to avoid. And this concept was initially developed for guidance of hysterectomies, which is one of the most common procedures performed with the Da Vinci robot. And in this procedure, it's a procedure to remove the uterus. There's a range of complications, medical complications that might require such a surgery. And in the removal of the uterus, we want to sever the major blood supply to the uterus, which are the uterine arteries shown in red. And we want to sever this blood supply while keeping the ureters, which are shown in yellow, intact in order to preserve urinary function. So our proposal is then to insert some contrast agent into the urinary tract in order to enhance the photoacoustic signal from the ureters. And so I'll show an example of how we did this in a cadaver model. And in this example, we see here a ureter filled with contrast agent methylene blue. We have an external light source and we have a tooltip. And we can provide information like the images that you showed, that I showed on the previous slide. And we can also, think about the burden on the surgeon and having to look at the surgery as well as the image. We could alleviate that burden by mapping the critical distance to a sound so that the surgeon can avoid the structures just by listening rather than visualizing the photoacoustic image. And so this is an example of how we can map the distance to a sound. So based on the input of the photoacoustic image, when the image shows that the tooltip is approaching the ureter that we don't want to image, we hear a sound that's played. And otherwise, the sound is not played, and we know that the surgeon is safe. And so we could see that there's a range of feedback that can be provided to the surgeon, both visual and auditory cues, in order to enhance the overall surgical procedure and improve patient outcomes. Moving on to a second type of implementation with the robotics, this would be what we call a visual servoing implementation, where we have an ultrasound system and a laser that is now introduced and connected to a robot arm that holds the ultrasound probe. And then the laser is connected to a catheter tip where we thread the optical fiber through a standard cardiac catheter in order to deliver light into vessel lumens or even the heart if we're doing a cardiac intervention, where the goal is to guide this cardiac catheter through the vein and toward our heart in order to administer some sort of heart therapy like ablation. one challenge with this procedure and why we want to introduce a visual servoing approach is because it requires fluoroscopy in order to guide that catheter to the heart. And when we use fluoroscopy, we introduce a range of challenges, such as challenges with the X-rays that are required to see the catheter tip. And this is not only a challenge for the patient, who may have several procedures over their lifespan, but also for the doctors who are using this technology on patient after patient, day after day. So we want to see how we can avoid this radiation risk by replacing the X-rays with a photoacoustic visual servoing system. And the visual servoing system is designed to track the catheter tip as it's navigated toward the heart. And so here we see an example of me holding a cardiac catheter at the bottom of this video, and I'm subtly inching it in and away from the heart, back and forth. And we can see that the ultrasound probe, which is held by the robot, is able to guide that catheter to, or is able to follow that catheter path, as you saw when the video was playing. And here's another example of that. And what's happening is basically the optical fiber is generating a photoacoustic effect that you could see, that we would see on this ultrasound screen. And this information is conveyed to the robot to find and stay centered on that photoacoustic signal. And ideally, this process would continue as the catheter is guided toward the heart. And once the catheter has reached the heart, as shown in this example, we can appreciate that while the fluoroscopy image does a great job at finding the catheter tip, and ultrasound image does so in some cases, but not in others, such as when the catheter is in contact with the heart, we can appreciate that the photoacoustic image provides a clear view of the cardiac catheter tip at all times, regardless of tissue contact. And this is, again, the same catheter tip that I showed for the visual servoing system can now be seen not only in the vein, but also in the heart. And it's very promising for replacing fluoroscopy and all the challenges that would be introduced because of the usage of X-rays. And so with this set of examples of implementation, we can appreciate that photoacoustic imaging is promising to guide multiple robotic and non-robotic minimally invasive procedures and surgeries. Over the past 5 or 6 years, we have introduced a range of techniques. I didn't have time to get into all of them, but it also begs the question as to what's next. And if we think about the years ahead since these techniques have been introduced, our goal initially was first to demonstrate feasibility for multiple surgeries and procedures. And our end goal is to commercialize these technologies so that they're useful for a wide range of patients all over the world. And if we think about what gaps exist in order for us to get to this commercialization aspect, we can appreciate that there are several new enabling and supporting research directions that I'll talk about in the next half of my presentation. One of those directions is laser safety for specific tissues. A second direction is the promise of flexible array transducers to conform and contort to different shapes and sizes. And the third is how we can improve our signal processing with the introduction of these newer research directions. So starting with laser safety and why it's a challenge, laser safety is only defined for skin and eyes. And I put a footnote for skin because this is something that's gotten really big in the optics field in the recent years, although it's a problem that has known to exist for decades. And in the spirit of Black History Month, I want to make it known that even though we have a limit for skin, there is no particular reference to how this limit relates to different skin tones. We know that optics technology is affected by skin tone. We saw that come to bear in the pandemic with the pulse oximeters and how there could be a disparity if your optics-based technology is not calibrated for different skin tones. Well, the same type of concerns would exist with this light-based technology when imaging through different skin tones. However, we are looking at these interventional techniques, and we're not so much suffering from the fact that there is no limit for different skin tones, but we are suffering from the fact that there's only limits for skin and eyes. And so our community is forced to use the limits for skin when we want to understand safety with respect to other organs. And what we see is that these limits that we use for skin as a proxy for limits everywhere else in the body, these limits are conservative when you're in contact with multiple internal organs, as demonstrated for the prostate, heart, and liver in the examples shown here. And so this is what led me and my lab to explore how we could expand the suite of guidelines with respect to laser safety in order to enable this new and emerging field of photoacoustic-guided surgery. And we started with livers, where we have a liver surgery setup and procedure shown here. And we have similar components that should be familiar to us from the beginning of this talk. We have an ultrasound scanner over here, a robot arm that can hold the ultrasound probe. We have a laser that delivers the light, and our subject is this swine that was imaged. And we followed a set of protocols and procedures as listed here with this equipment. We acquired color Doppler images to ensure we were looking at blood vessel locations. And the robot was used to hold the ultrasound probe in order to make 3D volumetric images. So this robot is very versatile. We saw before how it could be used to follow tooltips, which is true in this case. And now I'm about to show you how we could use it to make 3D volumetric images. So first, I want you to appreciate why photoacoustic imaging is useful. If I have a light source, which in this case, it's operating independently, but remember, it could be attached to a surgical tooltip, and I navigate that light source around my surgical space, I could see that when the ultrasound probe is fixed, I get subtly different signals, whether when I'm inside a vessel versus in the tissue surrounding the vessels. The signals are more focused when the light intersects a vessel, and they're more diffuse outside of the vessel region. And this is believed to be related to the presence of major vessels, which provide more focused photoacoustic signals, and minor vessels surrounding the major vessels that provide some level of signal, but the signal all blends together and provides more of a diffuse signal from these out of focus vessels. And here we see this major hepatic vein, confirming that this is the location, and it was confirmed with Doppler, as well as obvious in the ultrasound image as well. And so the benefit of the photoacoustic imaging is that as we're operating, we can determine where this major vessel is located so that we could say, target it if we want to cauterize this vessel or heat it up first so that it stops any possible bleeding. We could do that. Or if we want to make a cut and injure this vessel so that we could proceed with our treatment and avoid excessive bleeding and prepare for excessive bleeding, we can have that additional information where the vessel is located relative to the tooltip, and we would not be able to know that exact information with just purely an ultrasound probe or purely a Doppler method. And although we see this benefit, the challenge is that the energy that was used to achieve this benefit is greater than the limit reported for skin. And so this is one reason why I'm very keen to understand what are the safety limits for different tissues. And just to further motivate the technique before I talk about how we explore safety limits for liver tissue, here is an example of the 3D volumetric reconstruction that we can achieve when we translate the ultrasound probe across the tissue surface to build up this 3D vessel location. Again, if we want to target that vessel or avoid it in order to avoid excessive bleeding during surgery. So if we look at the profile of energy that was applied in order to get images over the span of the entire procedure and operation that generated the images that I showed you, we can see that we stepped up and stepped down the energy as we were testing around. And we stepped it up and stepped it down in order to see if this effect of distinguishing major and minor vessels was present at lower and higher energies, and we found that there was a limit at which we could see this distinction, but that limit exceeds the safety limit for skin. And so this led us to investigate whether the limit that we exceeded was actually damaging to the tissue or not. And so we took tissue samples from the liver before we did any experimentation. And that's what you see on the far left. That's our pre-experiment control. We also took tissue from a sample of the liver that we did not irradiate with laser, and that's our post-experiment control. And then we took tissue samples where we know we applied laser region, and that's our post-experiment laser region. And we could see differences in pathology. In particular, we see that where there was laser applied, we see some hemorrhage and degeneration and necrosis. And when we compare that to both controls, we don't see the same amount of necrosis. Necrosis is bad. We don't want to kill cells in the process of applying the technology. So this led us to question, did our technology actually introduce damage when used as intended? And I say when used as intended because we were initially just testing feasibility and stepping up and stepping down the laser energies even more than what we actually needed in order to achieve differences. So we returned and did another experiment, and we only kept the laser energy to exactly what was needed in order to perform a photoacoustic-guided procedure. And that's what this plot on the left shows. It shows our energy as a function of time. We see the pulse-to-pulse energy is very erratic. That's a common property of lasers. We can report our average energy over time. And we have a target energy that we were trying to stay within. And we have limits that we know we did not want to exceed on either side of the target. So this is an example of one sample over a duration of 10 minutes. And we did this for multiple time durations, 20 minutes, 10 minutes, and 1 minute. And we did this for two groups of swine. One was a non-survival group where we immediately, just as in the first experiment, sacrificed the swine and determined what type of damage, if any, occurred to the liver tissue. And the second group was allowed to live for 6 weeks before sacrifice. And again, we determined what type of damage. And the key thing to take away from this slide is that these box plots show the energy profile for the samples, the 3 samples from each swine. And we can see that, thankfully, as intended, a consistent amount of energy was applied to all samples within the range expected. And so we are now able to make comparisons across these different samples. And so here is a summary of the comparison that we published in 2021, where we see for the two groups, non-survival and survival, we got no necrosis. That is the most exciting result on this slide. We did get some other pathologies such as hemorrhage, inflammation, and fibrosis, which we can explain based on what's expected. For example, after a certain amount of time, the tissue does cause a fibrotic response. So we saw that expected response in the liver tissue in the survivor group, but we did not see it in the non-survivor group that was sacrificed right away. We saw some inflammation in the non-survivor group, which is expected just because the tissue had to be handled as the laser was applied, but that inflammation, the presence of inflammation went away from the survival group. And we saw some instances of hemorrhage across both groups, but we consider this also to be an artifact of the procedure itself and not necessarily something that was caused by the laser, which we are most concerned about, the laser causing cell death or tissue necrosis. So this is overall a positive result, and it shows us that although we did exceed the currently acceptable limits for skin, we did not cause any cell death by the laser. The laser did not cause any cell death. And this is an exciting result because our initial experiments, again, stepped up and stepped down the energy more than what was needed. And we also did the entire procedure for much longer than what we would expect to need the procedure, how long we would expect to need our technology for. And so the first experiment was over a duration of 80 minutes, where here we limited it to 1, 10, and 20 minutes. And we see that over that time period, we're safe. So this is very great and exciting for the whole field of this technology with respect to tissue-specific specific safety limits. And so now I'm interested in exploring how this approach can translate to other types of tissues. And so in the last few minutes, I want to now talk about this promising idea of flexible arrays in the context of photoacoustic-guided surgery. So imagine that this phantom that you see here is any one of the body parts that I talked about earlier in my talk, and see how the probe, it's not rigid, it could just wrap around the body part, the phantom body part, as we call it. And where if it was a rigid probe, we wouldn't be able to have this nice contact with the body part. And so here we have a video showing one of the first images from my lab, demonstrating the utility of this technology. And we could see how when the optical fiber is inserted here, we get the photoacoustic image that you see over here. And so this is very exciting for the entire field of flexible array technology to guide surgeries. And with this excitement, we can use it for addressing some of the challenges that we know that we introduce with the flexible array. So we like it because it can conform to different surfaces, but when it conforms to different surfaces, the array shape is unknown, and more broadly and generally for the field of photoacoustic imaging, sound speeds are also unknown. And so here's an example of an image of a photoacoustic target that we might get if we know the correct array shape versus if we don't know the correct array shape and we try to reconstruct anyway with a wrong value. We can appreciate how much distortion we get in the target, which is bad for localizing structures of interest. And so my group developed a new metric that we call MLOC, which I don't have time to go into how it's calculated, but the terminology stands for the maximum spatial lag 1 coherence with surrounding a target of interest, or within a target of interest. And so when we plot this MLOC value as a function of a radius that we guess because we don't know it, what we see is that the value, it peaks when the radius is correct. And so we could use that peak to say that this is our correct radius, and take that information and create an image based on the information provided. And this same metric can also be used to now estimate sound speed. So here's an example again of the MLOC metric now plotted against sound speed guesses. And we get a peak when the sound speed is correct. And that same peak value of sound speed is used to create the same image that you see over here. And this result is very exciting, not only for flexible array technology, but for the field of photoacoustic imaging in general, because all of photoacoustic imaging suffers from this unknown sound speed challenge. And so it's really exciting how we can get an accuracy range of 97 to 100% using this new metric, and also exciting that we can apply new types of signal processing techniques that we would otherwise not have thought about or not have introduced without this new type of technology coming onto the scene. Another type of signal processing technique that I'll briefly go into is how we could use a flexible array and use it for surgical guidance and take an acoustic-based approach to understanding which components generated the photoacoustic signals that we see. So here's a broad overview of such a signal processing type pipeline, where we would acquire our photoacoustic signal radio frequency data, and then we would first filter any incoherent noise with some type of coherence approach, such as the one that is the MLOC metric is based on. And then we can apply some IQ demodulation and get the power spectra of the components that were identified to create photoacoustic signals. And then we can stack the spectra and do some principal component analysis and build an atlas, and then use that on our test data in order to determine the components of the signals of interest. And when we do that, in this example and in these papers referenced below, we compare our method to the existing approaches. So our method is shown here. If we just use a single wavelength and identify the acoustic frequency spectra, or we use only two wavelengths, these two, and identify the spectra, and then compare to these other existing methods, we could see that just visually, qualitatively, we do a good job at getting it right and reducing the number of misclassifications, especially when we have two wavelengths. And this is exciting because some of the more standard spectral mixing methods can require as many as 15 to 20 or more wavelengths, which you can imagine would slow down the interventional process and information that we need to access in real time to understand whether we have methylene blue inserted in a ureter or some blood vessel such as the uterine artery that we want to target. And so if we look at the quantitative results, where on the x-axis we have the other competing methods that others have developed, and we end with our method for the two wavelengths, one wavelength on the x-axis and another wavelength on the y-axis. We see that we basically get, there's a range of combination of dual wavelengths where we basically get similar performance to the standard approaches A and B using only two wavelengths, and we achieved a better or comparable accuracy than that obtained with spectral unmixing methods, in this case requiring 12 wavelengths. So this is really great and promising for the technology, not only for flexible arrays, but also for photoacoustic imaging in general. So I'll end by saying this idea of photoacoustic-guided surgery has a lot of promise. It's been featured in several technical and media news outlets. I'm thankful that it's been the source of generous funding for my lab and generous recognition. And I draw your attention to this one YouTube video if you'd like to see some more of our technology in action. We also have an invited review article that I co-authored with my then graduate student, now assistant professor, and I encourage you to take a look if you'd like to know more about this topic. And one more recent direction that I'd like to draw your attention to, I alluded to it earlier, it's about, non-invasive imaging. Everything that I talked to up until now has focused on interventional, invasive, minimally invasive imaging, but this same technology applies when we want to image through the skin. But as I mentioned, we have some challenges with imaging through the skin if we don't consider the impact of skin tone. And so if you take a look at some intermediate skin tone, we see that we get this type of photoacoustic image with our conventional delay in some beamforming methods. And then with darker skin tones, we get more absorption at the skin surface, and we cannot see the underlying vessels of interest. What is exciting about the work that we're doing is that we can change our signal processing technique, in this case, use the short lag spatial coherence beamformer, and we can see signals of interest regardless of skin tone. And so this work has been picked up by CNN, and it's been a hot topic in the optics field in general. And so if anyone is interested in this field, I encourage more work in this area so that we can have more equitable imaging technology that benefits all patients. And with that, I'd like to end by thanking my large team of graduate students, postdocs, undergrads, and collaborators, and research engineers who have helped to make this work possible. I also thank my generous funding sources. CZI was highlighted. That's one of my most recent ones I'm very grateful for. And follow me on Twitter if you'd like to know more about the work that I'm doing in real time. I love to share and tweet, I guess, or X as it's called now, what we're doing. And thank you all for your attention. I'm happy to take any questions.
That was such a fantastic talk, really, really exciting. And it's an exciting way to start the series of this month. So thanks a lot for that. I loved your analogy of the thunder with photoacoustics. So I think, the presentation in terms of presenting, you're so amazing, and the work is fantastic.
Thank you.
You raised, so I mean, the side of equitable imaging technology, I think it's, so important for all aspects. I wanted to ask you regarding safety limits. So sorry, first, is there any question from the audience already? Maybe there is. Maybe, yeah, let's start with the audience. Holly has a question.
Hi, sorry, I actually have a lot of questions. Am I, my thing is glitchy. Hi, Dr. Bell, amazing work. I'm kind of blown away by how much you've done. And I have a lot of questions, but I'm going to go with the more personal one, because early on you mentioned guiding catheters using photoacoustics, and I'm just wondering if that's being done today in live patients.
Unfortunately not. It's still on the newer side of development, but hopefully one day it will be.
What do you think is, just a short follow-up, I promise you guys, for the surgeons or the doctors doing the procedure, do you think it sounds like, sorry, it'll be a pretty easy training, right? Because they can listen for sound and still see the image that they're used to. So anyway, you're just blowing my mind right now on so many levels.
Thank you so much. And as you said that, I don't think if my sound was playing, but when I showed the sound, hopefully you still got the connection.
Yeah, definitely. Thank you.
We have one question in the chat, which is from Carla Novel. It says, do the differences in skin tone translate subcutaneously to organs? And does dehydration affect signal capture?
So let me, could you repeat, or I can try to pull up the chat so I could see it?
I can repeat it. So do the differences in skin tone translate subcutaneously to organs? And does dehydration affect signal capture?
Okay, so yes, they translate to organs in the sense that organs still have some level of melanin, even though like skin is, has the highest concentration of melanin, especially in darker skin tones. But the expectation is melanin is one of the optical absorbers that we have in our bodies. It's not limited to skin. It's not even limited to humans. Zebrafish have it, other types of animals have it. And so it's understandable that different organs would have it as well. And then in terms of hydration, interestingly, water is yet another optical absorber that we have in our body. And so we do have to be careful about the wavelength that we choose, because based on the wavelength that we choose, we might have more or less optical absorption by water. We ideally want to have less optical absorption by water if we're not trying to image water. Similar to melanin, if we're not trying to image melanin, ideally we would want to choose a wavelength that has lower optical absorption of melanin. Thankfully for water, there is a window where we can achieve that, but melanin has such a high optical absorption across multiple wavelengths, which makes wavelength selection harder as the sole approach to address the challenges introduced when melanin is not taken into consideration.
Excellent. There's another question from Nikki. What are the challenges you are facing getting these technologies commercialized and available? That's as a follow-up to Holly's question. It sounds like laser safety is one. Are there others?
So several challenges that we are facing, because now, I am looking into the commercial direction, and one of the things is finding, partners for the technology who want to push it forward in this direction. And I raise that as a challenge because it really affects the business model. Like I have a knack for the software side of things, and short of me developing my own hardware to implement this, it would require some partnership with existing photoacoustic companies. And now these companies have to kind of have this idea on their roadmap. And so it's really kind of, I would say, kind of challenging for them to kind of shift gears sometimes if they already have other things that they want to pursue, to place this on the roadmap. So that's one challenge. And then kind of, breaking into different markets. There's photoacoustic imaging has historically been around for small animal imaging. And here we want, a technology for patients. So humans, that means FDA approval. So not a lot of the existing photoacoustic systems have that approval. In fact, there was one that was the first to get that approval just last year. So it's still very new of an area as it relates to patients. And so people are still kind of learning how it can benefit the patient market.
Just as a quick follow-up, is it difficult finding those companies because they they don't appear to be open to new technologies, or they just are fearful because there's so much sort of groundwork that still needs to be done around this? Because if this is happening in the veterinary field already, you know, most of the drugs go into the veterinary market and then they come into the human market. So clearly this is starting, but this seems like such a wonderful opportunity, because, you know, it's rather like having the cameras and the beeps on our cars nowadays when we're moving around. Having a surgeon have all of those clues when they're in there doing a surgery, to me seems like a total no-brainer. We should be doing it, right?
Right, I agree. I need you on my board of the company that will hopefully one day get started, so we could all kind of sell this idea together. For sure, I'm in agreement.
I think we are now at the hour, so I had several questions. I'm sure other people too. So I encourage everyone, if it's okay, to email you with these questions.
Absolutely, please.
And thank you so much. It was such a wonderful, wonderful talk.
And thank you again for having me. My pleasure.